Okay, so thank you, Laura. So I'm going to talk about the scoring and the results. So I'm going to focus mainly on sub-challenge 1 because the scoring procedure for sub-challenge 2 has similarities to that of sub-challenge 1. So sub-challenge 1A was network inference from the experimental RPPA data. So just a reminder, participants were asked to infer 32 causal networks specific to each cell line stimulus regime. But in this setting, there are no definitive gold standard causal networks which we can use to assess the performance of the inferred networks. But we do have this held out inhibitor data, or test data, which contains inhibitors which are not contained in the training data. So we use this to assess performance, and in particular, we use a novel held out validation approach which emphasizes the causal aspect of the challenge. And I'll now describe this approach. So we're interested in how well inferred causal networks agree with effects observed under inhibition in the held-out test data. And these effects observed under inhibition will be our gold standard. So the first step is to identify this gold standard, and we do this with a paired t-test. And we compare DMSO control time courses with test inhibitor time courses, and we do this for each phosphoprotein and each cell line stimulus regime. So for example, if we take UACC812 cell line and serum stimulus and test inhibitor test 1, we see in this example that under the test 1 test inhibitor, we see a clear decrease in abundance of this phosphoprotein relative to the control, and we get a small p-value from our paired t-test. Whereas in this example, for this phosphoprotein, we don't see any effect under the test inhibitor, and we get a large p-value. And we can do this for all the phosphoproteins, essentially ending up in a list of phosphoproteins that are the phosphoproteins that show an effect under the test 1 inhibitor. And this is our gold standard. And we can obtain such a list for each cell line stimulus regime and each test inhibitor. So now we have the gold standard, we can score our submissions. So participants submit a set of edge scores for each regime. And here's a matrix representing the scores for one regime. So we threshold this matrix to obtain a binary matrix, which corresponds to a directed network. We then look at the protein in the network that is the target of the test 1 inhibitor, which is the red protein, the red node. And we identify the proteins which are downstream descendants of the test inhibitor target, which are the blue nodes. So this is, and we can end up with a list of phosphoproteins which are downstream descendants of the target protein. So the idea is that these are the proteins that we would expect to show effects under inhibition of the target protein. So we can compare the descendants of the test inhibitor target with the gold standard list of observed effects in the held-out test data, giving us a number of true positives and a number of false positives. And then we can repeat this for varying thresholds tau, and this gives us an ROC curve, and then our metric is the area under this curve, so the AUROC score. Okay, so we have 32 of these AUROC scores for each submission, so corresponding to each cell line stimulus regime. And here we have the null distribution over the AUROC score for one of the regimes, and on the x-axis, these dots are the participants' scores, and the green ones are the ones which are statistically significant, which there are several, and the red one is the best performer for sub-challenge 1A, and that's their p-value. And so we have similar plots for other regimes, and we see that the best performer does consistently well in the ones shown here. So how do we go from these 32 AUROC scores to a final ranking of submissions? Well, what we do is we rank these submissions separately for each of the 32 regimes, which gives 32 rank scores for each submission. We then average these to obtain a mean rank for each submission, and this then gives us the final ranking. Okay, so we then want to verify that the ranking is robust, and in particular, that the top scoring team is robustly top. So we do this by masking 50% of the phosphoproteins at random in each of the AUROC calculations, and we then recalculate the final ranking and repeat this 100 times. And these box plots show you the 100 ranks for the top 10 teams. And we do a Wilcoxon-signed rank test to test whether the 100 ranks for the best team are significantly smaller than those for the second best team. And we obtain a small p-value indicating that they are, so the top team is ranked robustly top. Okay, on to part B, which is network inference from in silico data. So here, the procedure is more straightforward because we have the data-generating causal network, which we can use as the gold standard. And participants submitted edge scores, so they submit a single set. These are compared against the gold standard to get a single AUROC score per submission. So here we have the null for the AUROC scores for this sub-challenge. And we see that 14 of the 65 submissions were statistically significant. And again, the best performer had a highly significant score. And again, the robustness analysis shows that the top team is robustly top. So 59 teams participated in both Parts A and Part B, and we wanted to reward teams that showed a consistently good performance across both parts. And to do this, we calculated a combined score, which was the average of the final rank of Part A and Part B. And this is the red curve shown here. The blue curve is the final rank for Part A, the green curve for Part B. And we see that the team with the best average rank was also the best performer in Part B. And we also see that some teams that performed well in Part B did not all that well in Part A, and other teams showed the opposite performance. Okay, so on to sub-challenge 2, which is time course prediction from the experimental data. So if you recall this, participants were tasked with predicting phosphoprotein trajectories under inhibitions not contained in the training data. And these are assessed using the held-out test data. And in particular, we look at how well the predicted trajectories agree with the corresponding trajectories in the test data. And we quantify this using a root mean squared error, and we calculate one of these for each cell line, phosphoprotein, and test inhibitor combination. So just like in sub-challenge 1A, there were several AUROC scores per submission. Here there are several RMSE scores per submission. And here are null distributions for the RMSE for a single phosphoprotein under a single test inhibitor. So note that smaller RMSEs are better, and here we actually have two best performers. And we also have several teams which made predictions with RMSE scores which were an order of magnitude higher than scores you obtain from the random null. And we didn't observe this in Part B of this challenge, so this is interesting. It'll be interesting to see why this happened. And just as in sub-challenge 1A, we obtain our final ranking by ranking each of the regimes separately and then calculating a mean rank. So I just mentioned that there were two best performing teams here, and our robustness analysis revealed this. So the team with the top score was actually robustly top, but unfortunately this was an incomplete submission as they did not submit to Part B. So we looked at the second best team, but they were not robustly ranked second, but the third team was robustly above the fourth. So teams 2 and 3 are the two best performers. Okay, Part B, time course prediction from the in silico data. So here, the scoring procedure is essentially identical to Part A with a root mean squared error. Again, we have some nulls. So here, we see there are two of the 11 participants have significant RMSE scores, whereas in this regime, 9 of the 11 do. So we see there are some regimes which are easier to predict than others, and we also saw this in the other sub-challenges. And again, the robustness analysis revealed the top score was robustly top. As in sub-challenge 1, we calculated a combined score, and the score with the best combined the team with the best combined score was the best performer from Part A also. And again, we have teams that did well in one part and not so well in the other. So finally, on to sub-challenge 3, the visualization challenge. So there were 14 submissions, and 36 of the HPN-DREAM participants voted, assigning ranks 1 to 3 to their favourites. And these are shown in the bar plot here. And then the final score was the mean rank, which is the green curve. So we see that the best performer had a mean rank which was quite a bit lower than the next best. Okay, so to conclude, we think this challenge was engaged with well by the community, and we had a healthy number of final submissions, which were rigorously assessed using held-out test data. And in sub-challenge 1A, we used a novel procedure to assess network inference performance in a setting with no true gold standard. And there were many statistically significant predictions submitted across the sub-challenges. And here there are some areas that we'll be looking at. I won't go through them, but I shall leave them there for you to read, and I shall stop there. Thank you.